All right, hello, welcome everyone. Thank you for tuning in to Diagnostic's latest webinar. Today, our speaker is Dr. Alice Garrett from the Institute of Cancer Research in St. George's Hospital, who will be speaking on variant interpretation in cancer susceptibility genes. Here is a brief look at our agenda today. My name is Ian Moore. I am a network development manager with Diagnostics. I'm going to give a brief introduction about who we are as a company. I will then turn it over to Dr. Garrett for her talk, and at the end, we will dedicate some time to audience Q&A. At any time during the presentation, if you have questions, there should be a box below your screen. Feel free to type your questions into there, and then we'll be able to ask Dr. Garrett at the end. So Diagnostics is an end-to-end services provider and data analytics company, and our mission is better testing, better treatment. We're building towards a future testing ecosystem where all patients receive the right treatment at the right time. We believe that labs need to be involved earlier in the precision medicine decision-making process. We have experience in almost 100 disease pathways, and we've worked on every precision medicine brought to market with 36 of the top 40 pharma clients. And we also have a global presence across 53 countries, which enables knowledge sharing across a vast range of topics. All diagnostic labs, regardless of shape or size, can benefit from a free DXRX registration membership, and we view this collaboration as having a positive patient impact all over the globe. Through a free DXRX membership, you receive free access to easy-to-navigate US claims data dashboards, a curated feed of CDX and precision medicine news and resources, Diagnostics and pharma-sponsored collaborations for both labs and pathologists. In addition, you get more lab-focused webinars from speakers like the one that you have right now. So with that, it is my pleasure to introduce Dr. Alice Garrett. Dr. Garrett is a clinical cancer geneticist currently working as a locum consultant at St. George's Hospital in London. She completed her PhD in the field of genetic susceptibility to sarcoma at the Institute of Cancer Research in London, where she is now an honorary clinical lecturer working closely with the Cancer Variant Interpretation Group. Dr. Garrett, thank you for joining us today. And with that, I will turn.
Thank you so much, Ian, and thank you to Diagnostics for having me today, and thanks everyone for joining as well. So today, the main kind of learning objectives from today are to talk about variant interpretation in cancer susceptibility genes, and to compare that to how we might approach variant interpretation in other contexts. We're going to focus also a little bit on a possible approach to the interpretation of reduced penetrance variants, and think about how we also might approach variant reclassification, which is a particularly tricky area. And so I'm just going to sort of structure the talk as I kind of outlined before, to go through all of those learning objectives. But as well as the sort of a bit of the background information, I'm also about variant interpretation in general and how it pertains to cancer. I'm also going to do a little bit of an introduction to the Cancer Variant Interpretation Group UK that I do a lot of work with. And just before I continue, my conflict of interest is that I've previously done a sponsored webinar for AstraZeneca. So just to start off with, some general background to the variant interpretation in the germline, and just to be totally clear about what we're talking about today, if we think about the germline variants versus somatic variants, germline variants are those variants that are present in the gamete. Apologies. So they're present right from the first cell and form part of every cell in the body, including in the germline. And those variants can therefore be passed on to offspring. And if we compare that to somatic variants, particularly thinking, for example, in the context of cancer, these are variants which have developed sometime during a person's life and might be present only in a sort of subset of cells, for example, that of a cancer, and those variants are not passed on to offspring. So they're not inherited and they cannot be passed on. And today I'm going to be focusing just on the germline variant interpretation, and that's sometimes those variants are sometimes also called constitutional. So what are the things that we have to think about when we're doing germline variant interpretation? So, you know, we're usually arranging genetic tests because someone's presented with a particular phenotype. So we need to think about the nature of that phenotype, how frequently we see it, what's known about the genomic architecture of that phenotype, and how specific is the phenotype we're seeing. We also look at the details of the variant we've observed. So what type is it? Is it nonsense, missense? How frequently has it been seen in the general population? And what proportion of variants of that type are pathogenic? And then we might also gather additional bits of evidence or have access to different tools, for example, the results of functional assays or in silico predictive tools. And each of those bits of evidence will have a certain amount of discriminatory power to determine whether a variant is benign or pathogenic. And anyone who has had any experience whatsoever in germline variant interpretation over the last 10 years or so will be familiar with this rainbow-coloured grid from the Richards et al. Paper from 2015, which was the standards and guidance for the interpretation of sequence variants, recommendations from the ACMG and AMP. And just to sort of familiarise people who haven't seen this before, basically each of these boxes corresponds to a different type of evidence, and the ones on the left of that red dotted line are evidence towards benignity, and the ones on the right are evidence towards pathogenicity. And each of those different types of evidence is allocated a different strength, so supporting, moderate, strong, and very strong. And in that paper, they set out how you might combine evidence of different strengths to give an overall classification, which would be likely pathogenic, pathogenic, benign, or likely benign. And then anything that we're not able to get into one of those categories would be a variant of uncertain significance. And this was a huge improvement in terms of consistency between labs and probably even within labs. It was a sort of categorical approach rather than a sort of quantitative approach. And it was, you know, having to cover a huge range of different situations in terms of the context of and types of genetic tests and their scope. So it's quite difficult to cover every different scenario, and there were areas of subjectivity within this general recommendation. Just some sort of examples of evidence items within this framework, some of which I've sort of alluded to. So you can get very strong evidence potentially under the PBS1 criterion. If you've got a nonsense frame shift canonical splice, exon deletion or initiation codon variant. You might also get, if the variant falls within a mutational hotspot or a really important functional domain where we don't see a lot of benign variation, we might get moderate evidence under PM1 according to the original framework. And one of the criteria that can only be used in specific situations in terms of cancer susceptibility genes, but is more commonly used in a rare disease syndromic sort of context, is this PP4 criterion, which is about phenotypic specificity. And this is where we've got a well-defined syndrome where we're quite confident that the phenotype fits, and there aren't that many other genes that would fit with that. An example within cancer susceptibility is Gorlin syndrome, which is a syndrome associated with a high risk of basal cell carcinomas, but we also see quite specific phenotypic features such as jaw cysts and palmar and plantar pits on examination. So as I said, this was a really, really useful framework to help people sing off the same sort of hymn sheet, as it were, but there was still quite a few different areas where some additional specification was thought to be helpful. And there have been loads and loads and loads of publications which have further gone into this. I've just got a very limited number of snapshots here, some of which are about going into specific evidence criteria, other of which are focused on specific genes, and there's lots and lots of more that I haven't put here. One of the things that we do have to think about carefully is the fact that a lot of these different bits of evidence are not totally independent. So we do have to think carefully about whether or not it's appropriate to combine different types of evidence. And so the CanVIG-UK group, which I'll tell you more about shortly, has put forward some recommendations in this grid about times when perhaps we might need to be a bit cautious about combining different evidence items, or where it might be appropriate to, for example, downgrade one of those bits of evidence if used in combination with a type of evidence that's potentially overlapping. For example, some of the predictive tools with some of the sort of functional results can be overlapping. So that was sort of the background to the way in which people tend to, pretty much all labs, I think, across the world now really, are approaching variant interpretation. And as I said, it was pretty much a categorical approach where you had these different strengths of evidence and then combine them in a particular way using the combinations set out. But actually, it was found that this framework fitted quite well into a sort of a Bayesian type of model. And what that meant in practical terms was that we were able to convert these categorical evidence strengths into points that we could then add up to give a total which would assist us with reaching our final classification. And then we could potentially go in and sort of work out likelihood ratios and things for the strength associated with different types of evidence, which then would allow us to be a bit more quantitative in the way that we're applying this evidence. And so, according to this model, what is a supporting bit of evidence would be 1 point, moderate would be 2 points, strong would be 4 points, and very strong would be 8 points. And then on the benign end of things, you'd get negative points potentially as well. And then you'd add up all of the points that you had, and the sum that you reached could give you a final classification. So if it's between 6 or 9, for example, you get a likely pathogenic classification, and 10 or above will give you a pathogenic classification. And that I found, sorry, something was a bit funny coming up with my slides. So yeah, and I find that quite a helpful way to think about this. And we actually haven't had a kind of full update of that ACMG-AMP guidance since 2015, but there is a new one in the pipeline, and this is likely to include a more quantitative approach than the previous version. And one of the ways in which this can be helpful is in terms of moving a little bit away from that quite binary or limited categorical approach where you put your variant into one of these 5 categories: benign, likely benign, VUS, likely pathogenic, and pathogenic, and think of it more of a kind of continuous scale, really. In my opinion, it's particularly important for the variant of uncertain significance category, because clearly if you've only got sort of 0 or 1 evidence points, there's very limited information, and, the likelihood of that variant being pathogenic is actually still pretty low, even if it's technically uncertain significance. And if you compare that to a variant where you've got 6 points, and your probability of pathogenicity, according to this framework, is about 81%, and it's really on the cusp of being upgraded to a likely pathogenic variant. So I, as a clinician, would counsel that quite differently to variants in the sort of lower end of the spectrum, and we sometimes refer to those 5-point VUS as a sort of hot VUS, as opposed to a cool or an ice cold one where there's a lot less evidence. And actually, even within the likely pathogenic category, that corresponds to a probability of pathogenicity of between 90 and 99%. And, again, the likelihood of that variant sort of staying as it is going to vary depending on the strength of evidence you have available. So actually, I find it useful to know where a variant sits within that category when I'm counselling a patient. So that's sort of a background to variant interpretation in general, cancer susceptibility gene or not. But now I'll go on to think a little bit more specifically about cancer susceptibility genes. And there's a huge spectrum of different cancer susceptibility genes, which are associated with variable numbers of cancer types and penetrance. And penetrance is the proportion of people carrying a variant in that gene who go on to develop the phenotype, in this case, cancer. And there are some genes which are just associated with just one cancer or very limited number of cancers, other genes which are associated with a greater number of different cancers, and then those which are associated with cancer, but also broader syndromic features, for example, the Rowland syndrome that I told you about earlier. And I've just got a couple of examples here of the way we can think about susceptibility to a couple of the most common cancers, so breast cancer and colorectal cancer. And there's lots and lots of different germline genetic factors that are going to be contributing to someone's susceptibility to these cancers. In the bottom right of each graph, you've got your risk single nucleotide polymorphisms. And these are gene changes which are very common in the general population and individually don't really have a huge influence on risk. So you can see, so the allele frequency is on the x-axis, and they're right in the more frequent end of things, and the relative risk, so the actual effect size of those variants is along the y-axis, but so you can see it's pretty small, really. So individually, they don't cause a huge effect, but if somebody has lots of these SNPs, that can overall have a more substantial influence on their risk, and so might result in a sort of a family history with more cancers than you might expect, even when we don't detect a high risk or moderate risk cancer susceptibility gene. And then each of these genes has, each of these cancers, you can see in the graphs that there are more traditional cancer susceptibility genes, and as a rough kind of, the general trend is that rarer genes are associated with a greater relative risk. It doesn't always totally kind of hold true. There's obviously some genes you can see on the plot there, which are relatively common in the population, but are still associated with quite a high risk. For example, BRCA1 and BRCA2, which I'm sure you've come across before, are high risk breast and ovarian cancer susceptibility genes. And then on the colorectal cancer side of things, you might recognize some of the genes that cause Lynch syndrome, so MSH2, MLH1, and MSH6. So it's sort of the challenges in variant interpretation in cancer susceptibility genes are in part related to the nature of the cancer phenotype. So unlike rare kind of pediatric syndromes, these phenotypes are really quite common often, particularly breast and colorectal cancer, and therefore we get there's limited specificity and there's a high phenocopy rate both in the general population. So if we have a set of cases, they're not all going to be due to a high risk cancer susceptibility gene, and even within a pedigree we might see phenocopies. So by what I mean by that is within a family where there might be a high risk cancer susceptibility gene, say causing breast cancer, there might be an individual within, or several individuals within the pedigree who have breast cancer but have not actually inherited the familial variant. Another challenge is the late onset of a lot of these cancers, which means we might not know who within a family will go on to develop cancer and know whether to assign them as affected. Well, they wouldn't be affected at the time that we're sort of reviewing the pedigree. In addition, because these cancers may only occur after people have had children, they don't necessarily impact on reproductive fitness, and therefore the frequency of these variants within the general population is much higher than variants which cause people sort of very life-limiting and reduce the likelihood of reproductive fitness. And this, one of the other sort of impacts of this is that most of the variants in the sort of big hitter cancer susceptibility genes are inherited rather than de novo. So they are inherited from a parent, they're not occurring for the first time in the proband. I've already kind of mentioned penetrance above, but even people who carry high-risk cancer cell susceptibility genes, there will be incomplete penetrance, and not everyone will be affected by cancer, and it might be age-related penetrance as well. I'm sorry, I think I wasn't getting my slide to move on. 1 second. Yeah, I'm now on the right slide. Yeah, and there's also the challenge that these phenotypes may sadly be associated with death. So we might not be able to retrieve the DNA samples from the affected individuals and also get samples to assist with segregation evidence identification. So going back to that framework that I showed you before, and thinking about which of these evidence categories may or may not be relevant to cancer susceptibility, we can see that some of them, in particular that de novo evidence that I talked about, and some of the other evidence categories just become much less likely to be relevant in the field of cancer susceptibility genes. And there are a few others which may be kind of relevant in specific situations. As I said, the phenotypic specificity evidence item might only be relevant in particular situations. And likewise, segregation is a challenge because of the high phenocopy rate and the complete penetrance rate, although we are increasingly have seen the development of tools which can take that into account and give estimates of sort of quantitative estimates of the strength of evidence from segregation, taking into account what we know about the penetrance in individual genes. So again, that's a sort of a bit of a taster of the challenges that we see in cancer susceptibility gene variant interpretation, but there are some opportunities as well, and the fact that this is often kind of relatively common phenotypes can be an advantage. For example, we may have greater access to case data because there is more studies done and greater number of cases observed, so we can potentially get some statistical power from that. And also, because these diseases are often, and genes are often quite well studied, we're more likely to have access to high quality, effective functional assays and predictive information than some of the rarer or less well studied genes. So that was a sort of introduction to variant interpretation in cancer susceptibility genes in general. I'll now just give a little bit of an overview of the work of the Cancer Variant Interpretation Group UK that I work closely with. So this group started in 2017 and now has over 400 members from clinical services and laboratories across the UK and the Republic of Ireland. We have monthly meetings virtually where we do things like develop consensus recommendations for the UK. We might bring tricky variants and try and use them for learning and also to achieve consensuses. And we might also have educational presentations and updates from international groups. And these are the results of a survey, which is actually quite old now. It's done over 5 years ago when we had much fewer members, but about half of people were clinical scientists. It's probably a bigger proportion now, actually. And sort of the majority of the rest were clinical geneticists, but there were some other professionals involved as well. And it's a mixture of people who do cancer genetics as their predominant or exclusive role, and those who have a broader range of activities. And we've got a range of different experiences with different cancer types as well. So there's a lot of the sort of resources that we work on are available on our website, and so we've got, for example, the CanVIG-UK consensus specification, which is the UK approach in general to the interpretation of variants in cancer susceptibility genes, and this draws on international guidance. It sort of builds on that original 2015 ACMG-AMP framework, and also draws on additional recommendations that have been published since then as well. There's also gene-specific guidance, again, strongly influenced by the ClinGen variant curation expert panels, recommendations where available, but where not available, sort of often a kind of interim UK consensus approach to the interpretation of variants in these genes. And you can see there's quite a lot of genes that we've looked at now. And we've had a look at some of the main functional assays that are used for variant interpretation in cancer susceptibility genes, and assessed how might be the appropriate evidence strength to apply this evidence during variant interpretation. We've also got report templates which help labs across the UK to have a bit more consistency and not worry too much about, the nuances of wording if there's a kind of a national agreed approach. So we've got them for lots of different scenarios. We've also got a variant interpretation, a variant database called CanVIG-UK, which is run or has been developed by the three fantastic bioinformaticians and web developers that you can see on the right there. And there's also a YouTube tutorial about how to use this database if you want to have a play later. So this has variant level resources for single nucleotide variants. We don't have this data yet for insertions and deletions, but those variant types, you can initiate a discussion about those variants in the forum that I'll show in a moment. So some of this variant, and so we try and think quite carefully about the information that goes in there to make it as useful as possible for people working in diagnostic laboratories, pulling in key functional assays, presented in as useful, as clear a way as we hope as possible to be used directly for variant interpretation. And we've also got case data from UK laboratories. So diagnostic laboratories have to submit their data to the National Disease Registration Service, and then we're able to get this data from that service to present here. And then we've also got data from the UK Biobank and also from Nomad. And this is really useful for variant interpretation because we're able to do statistical analysis comparing the frequency of a variant in cases compared to controls, so cases within the UK diagnostic labs versus population controls at UK Biobank, and we're able to do that matching by ethnicity to ensure they're sort of comparable. And I won't go into the details here, but there is recommendations in the CanVIG consensus specification about how you might do those analyses. And there's also a discussion forum, which was previously only for the NHS diagnostic lab community, but is now open to international users from other diagnostic laboratories. And therefore, if someone's sort of struggling with a variant, they might put out a call to the other labs to see if anyone's seen that variant and has any additional evidence. And people will get an email alert, which may be standard or urgent. And then any responses will be stored against that variant, so you can log in and have a look at the discussion that's previously been had about that variant. It's also very useful in terms of if there's a reclassification, that a notification will be sent out to all the users within the community. So that is open to, as I say, anyone working in a diagnostic laboratory now. So I'm now going to move on to a specific example of a challenge that we've been trying to work out an appropriate response to within the CanVIG-UK community. And this is the interpretation of variants of reduced penetrance in high penetrance genes. And at the moment, our gene level penetrance estimates are sort of averages based on our observations of pathogenic and likely pathogenic variants, be that from segregation studies or case control data. But actually, that's almost certainly a bit of a simplification, and probably the true penetrance is actually different between different variants. and also even for each phenotype that might be associated with a given gene. And this is a bit complicated and tricky. So to date, we've sort of largely ignored it, to be honest, because it's difficult enough to work out whether a variant is or is not disease associated, and whether to classify it as likely pathogenic or pathogenic without actually trying to do a variant level penetrance estimate, which we just don't really have the data for at the moment. However, there has been some evidence previously, particularly, as I say, in these well-studied genes, BRCA1 and BRCA2, that there are certain regions of the gene where you're more likely to develop ovarian cancer, and other regions where you're more likely to develop breast cancer if you've got a germline pathogenic variant there. And because this is sort of becoming more and more of an issue, sort of internationally, and the ClinGen group have commissioned a working group to sort of have a think about this, and this is a figure from one of their guidance documents, and it's sort of similar to those graphs that we looked at the start, thinking of the sort of relative risk associated with a variant. So right at the bottom, we've got those risk alleles, which have a very minimal effect individually on risk. And then right on the right-hand side, you've got your high penetrance variants, which are contributing in a greater way on an individual basis to a given risk. And then we've got these variants in the middle, which this group from ClinGen are calling low penetrance variants. And then within each of those groups, you can then have your confidence about how likely you think the variant you're observing is sort of truly associated with the disease. So you've got both your confidence about its association with disease, and then the sort of estimated effect in terms of on risk of that disease. And we're still sort of simplifying things by putting it into three categories, but that's because it is going to be a kind of continuous spectrum, but this is the sort of a more feasible way of kind of attacking the issue. And so within CanVIG, as I say, laboratories have been struggling with variants which they're a bit suspicious. They could have a bit of a reduced penetrance compared to their normal variants. And we have we slightly changed the terminology that we use. We've been calling these reduced penetrance variants because, if you start off with a very high risk, your risk actually might not be low, but it is reduced compared to a very, very high risk, for example, associated with BRCA1. And then we've got standard penetrance variants, which are having the penetrance and the risk of disease that we have been, quoting to our patients for years now. So we were thinking about how we could modify this framework compared to the standard penetrance framework that we use, and we started with BRCA1 and BRCA2. And there is one kind of poster child of reduced penetrance in cancer susceptibility genes, which is this R1699Q variant. And the Enigma consortium, which is a huge international consortium looking at breast cancer susceptibility genes, in 2012 published some data indicating that this, they'd worked really hard to gather data from all over the world and demonstrate that this variant had a lower penetrance than a typical BRCA1 variant. And they published some updated data in 2018 demonstrating that this in a bit more kind of granular sense. So you can see that red line is the penetrance for a typical BRCA1 variant. The green line is the risk in the general population, and the blue line is this R1699Q variant, and you can see basically it's in the middle for both breast cancer and ovarian cancer. So it is associated with increased risk of cancer compared to the general population, but not nearly as high as a typical variant. And the Enigma group have also made some recommendations about how that might change the management of people found to carry this particular variant. And essentially you might start the breast screening a little bit later than you might do for other BRCA1 variants, think a little bit more carefully about whether it's appropriate to do risk-reducing mastectomies, and also the age or whether to do risk-reducing salpinguvectomies for the ovarian cancer risk. But unfortunately, we are very rarely in the luxurious position of having all of this beautiful data to give a variant-specific risk. So we did it, and we sort of heard mutterings from the community that people were struggling with this. So we decided to do a little bit of a survey to see the scale of the issue. And essentially, so we start off by asking people how frequently they encountered this issue. And there was a variety of responses. The most common response was 2 to 6 monthly. We asked people how confident they felt in terms of classifying variants of reduced penetrance. So this is members of the CanVIG-UK community, predominantly clinical scientists in NHS diagnostic laboratories. And essentially, as individuals, the vast majority of people said they weren't very confident at all in doing these classifications. GLH stands for Genomic Laboratory Hub, which is the sort of regional laboratory group. So in collaboration with their sort of local and regional colleagues, people felt a bit more confident. But if there was a national discussion, people said that they felt a lot more confident. And everybody who responded to the survey said that they thought that some CanVIG guidance would be very valuable or extremely valuable. So we thought it was worth pursuing. And then so we thought about how we might approach this, and the evidence for and against reduced penetrance might not fit well within the standard framework, which is designed for sort of standard penetrance variants fitting the sort of mold of what we'd previously kind of described as typical features of this gene. And some variants which have historically been tricky to classify, for example, because they've got conflicting evidence, it's possible that they are, in fact, reduced penetrance variants. And then we thought about what are the sort of types of evidence that might help us work out what's going on in this context. And one of the key bits of evidence is obviously case control data, because that's where you're sort of quantifying the effect size and the risk associated with that variant directly from the data. And so, for example, if you have a high penetrance breast cancer susceptibility gene, such as BRCA1 and BRCA2, you might expect the odds ratio to be above 4. But if you've got a reduced penetrance variant, you know, how would you define that? It could be an odds ratio of 2 to 4. So it's still associated with some risk, but not in the magnitude that you would typically get with a high risk cancer susceptibility gene. And there's also other evidence types which might give you a clue. For example, you might see intermediate results on functional assays or conflicting results. You might get some leaky splicing if it's a splice site variant. And you might also get some clues from the phenotype of people carrying that, observed to have that variant. So quite apart from the sort of, you know, the numbers of cancers, for example, you're seeing in a pedigree. You know, if it's a reduced penetrance variant, there might be lots of people in the pedigree who carry the variant but aren't affected with cancer in the same way that you'd normally expect. And you can also potentially get clues from people who are homozygote for that variant, because some genes in the heterozygous state cause an increased risk of adult onset cancer, but in the homozygous or compound heterozygous state, we would expect to see a more severe phenotype, which might have, for example, childhood onset, such as Fanconi anemia, which is caused by homozygosity or compound heterozygosity in a number of different genes, including BRCA2. So you might potentially, if it were hypothetically a reduced penetrance variant, you might see somebody who didn't have classic Fanconi anemia, but perhaps had sort of cancer at a younger age than average, or very mild features. So this was obviously quite a challenging issue to tackle. So I won't go through all of these different stages, but the key point is that we kind of did various surveys, polls with the CanVIG community at our meetings. People were very patient. We kind of drafted a framework, applied it in some different some exemplar variants, updated it, and then eventually sort of put forward our proposed initial approach to this issue. And this is just an example of some of the scenarios that we asked people to think about. And this is, we did look at different types of evidence, such as the functional side of things, but this is with regards to case control evidence. And we just sort of presented different odds ratios with different confidence intervals, etc., and asked people how they would instinctively imply this evidence. And essentially, there was quite a lot of variability in people's responses, which sort of further demonstrated the fact that we needed to work together to come to a consensus. Otherwise, there was going to be huge inconsistency across different laboratories, and people wouldn't feel sort of very confident with this. So after all of those different steps, we sort of produced this framework. And the key thing to note here really is that there are two different scenarios that we envisaged. One, which is outlined in the red box there, where we had quantified active evidence of an effect size which was reduced compared to a typical variant. And this would usually come from case control data, but potentially could come from a segregation tool which was applied using a reduced penetrance framework. And then we've also got additional sort of specifications about how you might deal with different evidence types that could potentially indicate reduced penetrance, and also situations where you might want to think about being a bit more lenient about allowing evidence that would normally be considered to point strongly towards benignity, but actually could potentially be consistent with a reduced penetrance effect, for example, to do with the frequency of a variant in the population. So that situation is the one that we are kind of a little bit more comfortable with, because we do actually have that active evidence and that there is a reduced penetrance effect, either from segregation data or more commonly case control data. We've also got the scenario, which is actually more common, probably, where we don't have any active sort of statistically significant evidence of a reduced penetrance effect, but we're just a bit suspicious because there's a bit of inconsistent evidence, or the phenotype doesn't quite fit, and this could potentially be indicating reduced penetrance effect, even if we don't have quantitative data to sort of prove it, as it were. And this is a bit more, people in general felt a lot less comfortable with this situation, and in reality it is quite difficult to kind of confidently designate a variant as being of reduced penetrance if you don't have active kind of data to prove that. But this hopefully helps people to kind of potentially be able to assign that classification if there is lots of evidence indicating a reduced, or indicating that a reduced penetrance effect is possible. And this is just to show that try to keep it as simple as possible, but there are the inevitable caveats. So if you want to read more about this, it is published in Genetics and Medicine and also available on the CanVIG website. And we hope to review this in a year or so to see how people have got on with applying this framework. And so we've asked people to flag any variants where they're thinking about reduced penetrance on the CanVIG diagnostic forum using this box. And as I say, this is very much sort of just a starting point to try and improve consistency, and will be iterated and reviewed going forwards when we see sort of how it whether it works sort of in practice. So that's sort of reduced penetrance. That's all I wanted to kind of say about that. Another very, very challenging situation is variant reclassification. So as you can see, there's guidance and approaches to variant interpretation is very complicated. And you can see how even just a small kind of change in the evidence that becomes available, or perhaps an update to the guidance, can result in a variant changing, for example, from likely pathogenic to variant of uncertain significance, or vice versa. So this doesn't, this isn't a specific issue to cancer susceptibility genes, but I think we're probably seeing it a bit more often because the guidance updates are perhaps a bit more frequent, and the frequency with which new data becomes available is also perhaps a bit more regular. So we went through a sort of similar process for developing a CanVIG consensus approach to this situation. As I mentioned, for the reduced penetrance approach, again, lots of surveys and polls and discussions and iterations of the framework. This is sort of a different way of presenting the kind of quantitative approach to assigning an evidence, variant classification, as I mentioned before. And you can see that red vertical line is really the critical point at which a variant switches from variant of uncertain significance, where we don't do any testing in the family, we can't offer any screening, etc., to it just tips over from 5 points to 6 points, and then suddenly it's likely pathogenic. All of their relatives can get tested, people can have risk-reducing surgery, they can have prenatal tests. So that sort of threshold is really quite what we call the actionability threshold, is really critical here. And you can see how variants within that red box, so within the sort of 4 to 7 point mark, you can see how it's not inconceivable that they could go up and down across that threshold, depending on the guidance, the evidence. A new functional assay could come up, and that creates all sorts of challenges for scientists, clinicians, and of course the patients, for whom this is hugely anxiety inducing and makes their decision-making process really, really difficult. So, as I say, we went through all those various sort of stages, and this was another framework that we've put forward as, again, a starting point. It's led by Lucy Leung, my colleague at the Institute of Cancer Research. And the things that we take into account are the nature of the evidence that's resulted in the change in classification, the change in the score, so how many points have kind of have been added or subtracted to cause that change of classification. Obviously, whether it's an upgrade to, for example, to likely pathogenic from VUS or vice versa, and what the new classification is. And for each of those, suggested the approach that the laboratory might want to take in terms of when they issue reports, when they alert other laboratories, etc. And also the clinical team, the situations in which it might be appropriate to proactively contact families that have been seen historically, what to do about new families, and also relatives of historic families. So this doesn't make this problem any easier to deal with when you kind of encounter it in real life, as I have found recently. It's still really, really stressful for everyone involved, but most importantly, of course, the patients. But at least this does allow some sort of slightly more consistent approach and makes one more confident that one is doing something which is consistent with a national approach, and that it's likely that other services would do a similar thing if they encountered an equivalent scenario. And just to highlight that the rows in red are those situations where you've got a variant which is of potentially at the sort of at the threshold of that actionability kind of threshold, and it could sort of tip either way. We call that sort of potentially changeable classifications, and those obviously need to be dealt with particularly sensitive, because these variants bouncing backwards and forwards between likely pathogenic and pathogenic cause all sorts of issues for everyone involved. So this approach hopefully is aiming to be a bit more sort of sustainable and standardized, and allow appropriate use of clinical resources as well. And again, very much a starting point, and will be iterated as we see more and more situations where we need to think about this issue. And again, this has been published in Genetics and Medicine and is also available on the CanVIG website. So I just wanted to say a huge thanks, just to finish off with, to everyone who's been involved in this work. So the Professor Claire Turnbull at the Institute of Cancer Research, from whom I've borrowed several of these slides, in fact, and the program manager, Beth Tor, Sophie Allen in the team in particular, who does vast amounts of work for CanVIG-UK. We've also got a fantastic steering committee group, CanVIG Steering and Advisory Group, who puts in a lot of work and attend a lot of meetings to help with all this work. And of course, we couldn't do any of it without the CanVIG-UK membership, our national and international collaborators, and Cancer Research UK, who funded the CanGene CanVAR programme, which has funded a lot of the work of CanVIG-UK and other activities within the group for a number of years. So hopefully, I'm not sure I shared this slide at the start, but these were the objectives that I was hoping to cover, and I hope that you've found this interesting, and I'm happy to take any questions that you may have.
Thanks, Dr. Garrett. Definitely interesting, engaging. Really appreciate you sharing your experience and insights with this. So we do have some questions. The first one we'll lead off with is, could you elaborate on the specific challenges you faced during the iterative development of the reduced penetrance classification framework, and how were these addressed?
Yeah, so I guess it's always the same when, trying to reach any kind of consensus with quite a large group of people working in with different experiences and different perspectives in terms of trying to come and trying to, I guess, create a framework which largely works for the majority of situations. It's sort of, so the way because you're always going to come up with situations where it's like, oh, well, if we, there's two options, we choose this way, it works for this variant. If we choose this option, it works for this variant, but not this one. So it's often kind of trying to reach some sort of pragmatic agreement, which isn't going to be perfect for all scenarios and isn't going to fit in 100% with everyone's prior kind of thoughts, but most people largely agree seems reasonable. So the way we did have quite a few exemplar variants, and there was definitely one. So the sort of the first main kind of draft that we shared with the community, it worked for a couple of the scenarios we had, and there was pretty good consensus that everyone was happy that it worked and we could classify the variant using that framework as reduced penetrance. But there was another scenario where there was some conflicting evidence, and it was very much a kind of almost a 50-50 split in terms of where people were at. So we did kind of take that into account and redraft the framework and bring it back with some new exemplars. And we still didn't reach 100% consensus, but we kind of got, the majority of people agreed that that was probably kind of an appropriate way forward. So, I guess it's sort of doing the best, taking input and reviewing the sort of the available options. But if no option is perfect, just going with, a first offering with the acknowledgement that we will definitely be kind of iterating this and accepting that it's not perfect for all situations. I hope that answers the question. For quite a long answer.
Well, it makes, yeah, 100%. Thank you. So the next question is, how did you determine the appropriate evidence strength adjustments for potentially contradictory or weakly pathogenic evidence in scenario B variants?
Yeah, so this was really, really hard, and kind of it's sort of a circle where we don't have a good truth. If we had a good truth set, we could kind of try and quantify this evidence in a more objective way, and that is what we would love to do. But at the moment, the only real proper kind of exemplar variant is that R1699Q. So we just didn't have that kind of truth set with which to do the quantification, and this is why we had so many kind of loops of sort of discussion and surveys and votes, because we were trying to kind of take input from, we're extremely lucky to have a wealth of experience amongst the community and some really, really knowledgeable, experienced people. So kind of taking all of that insight to make what seemed like a reasonable starting point. But in all honesty, I don't know if we've had any variants. There's lots of ones that have been kind of put forwards as potential candidates for reduced penetrance under that scenario B situation, but I don't know if we've been able to kind of confidently give them the likely pathogenic classification, because obviously we're a lot more cautious in that situation. So it's quite, variants have to earn quite a lot of evidence before they're able to get that assignation.
Mm-hmm. No, thank you. So this question kind of just goes for the general framework that your team and your group uses. So what are the key considerations for applying this framework to other disease areas beyond high penetrance cancer susceptibility genes?
Do you think that's the consensus? Is that, I presume that's pertaining to our main cancer susceptibility? Is that the reduced penetrance or the main?
The question, so I think the question is asking, sorry, you know what, let me go here too. So I think the question is also just getting at, let me put it in there, there you go. So we have we have this framework that you've discussed, and what are some considerations for applying this framework to other disease areas beyond the high penetrance cancer susceptibility genes?
So it's very much focused on cancer susceptibility genes, but I've used high penetrance genes as most of my exemplars, just because it's sort of a bit easier to kind of conceptualize. But we would, in the family, include all of the moderate risk cancer susceptibility genes as well. And there is some, there are gene-specific guidance to cover a lot of those genes. In particular, the kind of moderate risk breast cancer susceptibility genes, and there are mention of sort of a moderate risk context in the main consensus specification. There are a lot of the paradigms which would, I think, apply to various other situations. In particular, kind of there's a lot of overlap with cardiac genes, so that those causing a high risk of inherited cardiac disease, lots of issues with phenocopies and incomplete and age-related penetrance, etc., and also some of them are sort of common diseases. So a lot of it could be applied, but and we do kind of have contact with with people working on the cardiac side in the UK, but our kind of remit and our community is focused on cancer susceptibility genes predominantly.
Mm-hmm. So it looks like we have time for one more question. What steps are being taken to validate the framework in real-world clinical settings, and how do you plan to update it based on feedback and new data?
So I'm presuming that is relating to the reduced penetrance, because I kind of presented a few frameworks.
Yes.
I would say, so that's why we're asking people to flag in Canva when they're thinking about this, so that we can then pull all of those responses and see what people have done, how it's performed. And we also want to know when people have tried to apply the framework, and even if they haven't kind of reached a likely pathogenic classification, whether they kind of felt that what evidence they were and were not able to apply, and how it kind of pans out in real life. And then we will, so it's kind of reactive, because we did at the upset, right at the start, we did try and gather potential exemplars to have a look at, looking at variants which had achieved, intermediate functional assay scores, for example, or from our case control data looking like it might have an intermediate effect. And then we did use some of those for our exemplars and our polls and things. But that is not the same as actually observing a variant in a diagnostic case in a real patient, and having to make an actual decision about how you report that variant and manage the patient and their family. So we thought it would be really helpful to actually get, see how this works in the real world. And we do plan to do that, used with the help of Canva. And we're lucky that we have this quite tight community that will kind of give us quite granular kind of information about how things have gone for them.
Awesome, thank you. Well, that is our time for today. Thank you so much, Dr. Garrett, for taking the time to speak with us, present on this. Thank you for everyone in the audience for taking the time watching this. Stay in touch with Diaceutics as we put on more webinars like this, and we hope everyone has a great rest of their day. And thank you again, Dr. Garrett.
Thank you for having me, and thanks for everyone for coming.